And we are now live. So welcome to week 2 of plant physiology. During tonight's class, we're going to cover some of the important concepts, starting with our first lecture, which is actually lecture 2, root structure and function. So let me just skip ahead to slide 18. Okay, and we're going to have a look at the structure of the root and make sure that everyone's really comfortable with this, and I'm sure you would be, because we covered this in a little bit of detail last year. So let me just grab a pencil and we'll start down here with these cells referred to as the root cap. And can anyone tell me what the substance is that's secreted by the root cap cells? What is the substance secreted by the root cap cells? And the answer is actually here on this slide. And if you don't know, just say no idea. Mucigel, perfect. There it is there, mucigel. Okay, and that's supposed to be this grey region that you can see around here. Okay, so that's the mucigel. Now let's focus in on this region of cells here called the quiescent centre. And what's the story here? Who knows something about the quiescent centre, this group of cells? Who knows what these cells are? What they are, what they do, what they don't do? No idea? Anyone else? You can just say no if you really don't know. Quiescent centre. Very strange region, I think. No idea. Okay. Birth cells. yeah, not quite the right way to put it, but yes, you got the right idea. Birth cells. They're actually undifferentiated cells. They're like stem cells, if you like, but they seem to be quite dormant. There's not a lot of activity happening in there, and there's not a lot known about this region of cells, but they do start to become activated in some situations. So for example, if there's damage to the root tips and cells and so on. So quite an unusual region. All right, now let's look at our 3 major zones. And of course, we've got the meristematic zone. And what's happening in the meristematic zone? Obviously, it's a region of rapid cell division. So that's pretty straightforward. So in this region, the cells, most of the cells haven't quite differentiated. They haven't taken on their final form and function, if you like, undifferentiated cells. Then we move on to the elongation zone. Okay, which is about in this region. And these are the cells that were formed most recently, and they're still growing, differentiating, elongating, and of course they're the ones that actually give length and growth to the root itself. And here we can start to see the differentiated cells within the actual root. And then finally we have the maturation zone, or zone of cell differentiation. And this is where the cells are all functional. The tissues are functional and the cells are functional. All right, so root hairs. What cell types do the root hairs arise from? What cell types do they arise from? Endodermis? Not quite. Good guess, but not quite. Yes, that's it. Epidermis. So, of course, your outer layer of cells here, usually just 1 cell layer, sometimes 2 cell layers. Some of these cells just elongate. Here it is. This is 1 epidermal cell that is elongated into a root hair. Sorry, that's a bit messy. Here's one epidermal cell there, and you can see it's just elongated to form a root hair. Okay, good. Then we have the cells in this region here, and these cells make up the ground tissue of the root, and we refer to those cells as cells of the cortex or cortical cells. Now let me just clear all that because it's already getting messy. Okay, now the coloured region in the middle, in the actual centre of the root, what do we refer to that central region as? That you can see there. That central cylinder. What do we call that? And you can see it's not quite formed completely down here. Vascular tissue, yes. So this is the central core vascular tissue within the actual root. And of course, that's very different to what you see in the stems. The stems don't have that central core of vascular tissue. They have several distinct vascular bundles right around the inside of the stem. But in the roots, you only have that central core. So I'll just clear that. All right, now let me find it. Even with my glasses on, this is a little bit tough to see where these lines are going. Where are we? Okay. I don't think it's actually labelled here. Xylem phloem, endodermis. All right, so everyone's familiar with this. Everyone's happy with the endodermal layer, the endodermis, which contains or is surrounded by the Casparian strip. What does the endodermis do? So single cell layer called the endodermis that surrounds that vascular, that central vascular bundle. Does anyone know what the endodermis does? Got someone answering in the background at home. Endodermis. What is the function of the endodermis? Yes, it does. It does separates the cortex cells from the pericycle layer. Yep. It certainly does that. Separates transport cells. Yeah, it does separate the cortex from the green layer of cells that you can see to the inside of that endodermis. Okay, so the clue is that the endodermis cells are surrounded, not on all sides, but are surrounded on certain sides of the cells by that waxy layer called the Casparian strip. That's the clue. That's the clue. No? Ghosts. Very good. I like that answer. All right, we're going to come back to that. We will come back to the endodermis, but as long as you can see where it's actually situated. All right, terrific. Well, let's, since someone's mentioned the pericycle layer, and you can see the pericycle to the inside of the endodermis layer, so the green cells. Any idea what arises from the pericycle cells? And it's not ghosts, Lachlan, even though I really like that answer. Branch roots, yes. What should we refer to branch roots as? I know that's been mentioned somewhere in the lecture, branch roots. Does anyone know the correct name for branch roots? Lateral roots, good. Okay, lateral roots arise from these pericycle cells. As you can see there, they would push out, crush these cells in here, and then emerge from the side of the root to get these lateral roots. All right, terrific. Let me clear that and, all right, move on. I've covered everything there. All right, let's move on to slide 33. And who remembers their chemistry from last year? Here we go. No, you cannot escape chemistry when you're studying plant physiology. You can't escape it. All right, so I know you all remember that water is a polar molecule. You know that water molecules form hydrogen bonds. You remember the concept of electronegativity, which means that you remember also that oxygen is more electronegative than hydrogen. So of course it attracts the electrons within that covalent bond between them. All right, now here's a good one. The geometry of a water molecule is tetrahedral. Yeah, what do we mean by that? How do you know when a molecule, the geometry of a molecule is a tetrahedron? Who remembers the rules of determining molecular geometry? Does anyone remember that? So looking down here might give you a clue. You've got a central oxygen, and how many groups have to surround that? Not quite the charge, no, how many groups are surrounding this central oxygen, which enable it to four groups? Thank you very much. So look down here, central oxygen surrounded by 1, 2, and remember, lone pairs of electrons also count as groups. So there we have 4 groups surrounding the oxygen. That's when you end up with the molecular geometry of a tetrahedron. But if you remember back to your previous studies, you might recall, and let me just clear that for a second, you might recall that when some of those groups, for example, here we've got two of the groups being lone pairs of electrons, that would change the bond angles. So even though, yes, we say that a water molecule forms a tetrahedron, but because 2 of those groups are electron pairs, they essentially compress that tetrahedron, and the shape, the molecular shape, is actually that of a bent molecule. So just a little bit of revision there for you. So it actually takes on less, the bond angles are sharper rather than a typical tetrahedron. All right, so a bit of revision from last year. Now let's look at another couple of concepts here. And I'll just clear that. Very good. All right, so we say that a water molecule has 2 positive and 2 negative poles. And who can point out those positive and negative poles? And I think, Lachlan, you were referring to it a little bit earlier. So if we look at this water molecule here, can everyone see? Yes, absolutely, positive pole here. And here's a positive pole here, delta positive, and here we have a negative pole. So in other words, we've got poles here, and we've got the positive and negative here, which is what that's referring to. Okay, let's clear that. And of course, that electrostatic interaction between the negative pole of the oxygen and the positive of a hydrogen on a neighbouring water molecule is what we refer to as a hydrogen bond. Now, you might recall also that hydrogen bonds alone are actually quite weak interactions between molecules. But of course, because there are so many water molecules together, and each of those water molecules can form up to 4 hydrogen bonds with neighbouring water molecules, then it becomes quite a strong interaction. So given that we have all these interactions between all these water molecules, what property do we refer to that as when we're talking about water, when we're talking about properties of water? That ability for water molecules to actually form all these interactions with each other. Oh, and we have a question here. So I'll just move that out a bit while you're thinking about my last question. Every oxygen forms hydrogen bonds with 2 other hydrogens. Yes, that's correct. Absolutely. So we can see that here, where the lone pair of electrons is here, then we can have an interaction here with a hydrogen from another water molecule, and the same thing can happen here. Yep, absolutely correct. And then, of course, we have, yep, this one here, and there's our 4 hydrogen bonds that each water molecule is forming. All right, so that ability, as I was saying before, that ability of water molecules to actually form all these interactions is described by the property of water called cohesion. So you might recall somewhere from last year that we talked about a theory of water movement in tall trees called the cohesion-adhesion tension theory. And that's where that term cohesion comes from, the cohesive nature of water molecules. All right, so we've covered that. Now let's move on to slide 35. So as a result of those adhesive and cohesive properties, hydrogen bonding gives water very high tensile strength, incredibly high tensile strength. And of course, this also is because of the nature of the xylem vessels being so incredibly narrow, and the capillary action that accompanies the movement of water. But we won't talk about the movement of water up the xylem just yet. We'll save that for next time. Okay, so you would have looked through this and listened to the lecture, and you know that pressure is measured in pascals or megapascals. This is what you'll see most frequently used, this particular unit here. And you already know about the 2 different pressures that exist in plants: positive hydrostatic pressure and negative hydrostatic pressure. So let's just recap. I'd like to make sure that you're understanding what these pressures are referring to, what they mean, and where they're actually found within the plant. So let's start with this one, positive hydrostatic pressure. And using this syringe as an example, I want you to explain to me how you would demonstrate positive pressure using that syringe. What would you do to demonstrate positive pressure? Push it. Very good. Push the plunger. Excellent. Good. So everyone remembers that, or you actually pick that up from the lecture itself. Push the plunger in, and of course the actual syringe is capped. There's no needle there. So that from that you can understand that positive pressure is a pushing force. Now, I want you to tell me, where do we find positive hydrostatic pressure in the plant? What part of the plant? Pushing force. Oh, yeah. Roots, absolutely correct. So the roots are under positive hydrostatic pressure, pushing force. Don't you love my writing? Excellent. Okay. So what would happen? This is something interesting too. What would happen to this air bubble? If there was an air bubble in that water, what would happen to it as a result of that positive pressure or that pushing force when you push in on the syringe? What do you think would happen to that air bubble? It would be compressed, exactly right. It would actually become a little bit smaller because of that type of pressure that's being exerted. Excellent. All right, now let's look at this one, negative hydrostatic pressure. Tell me how you would demonstrate that using the syringe. How would you demonstrate negative? Yes, absolutely right. You would actually start pulling back on the actual plunger. Yes, pull back on it. So what sort of force is that creating? So remember we said positive is a pushing force. What sort of force is that creating to give us the negative pressure? Rather than pushing force, it's a pulling. Good. Suction, yes. Pulling or suction. Absolutely fantastic. Now, where do we find, and that's supposed to be the word pulling, where do we find negative pressure in the plant predominantly? Stems, definitely. So up the xylem, in the stems, definitely, and the leaves, the xylem in the leaves too. Absolutely. Okay, good. Excellent. So everyone's clear on pressures. Okay, just one more thing about pressure. Think about a cell, a plant cell, and that cell, oh, I shouldn't have drawn it like that, sorry. I should draw it like this to be a bit more accurate, with the cell walls slightly extended. They're the cell walls, and there's your plasma membrane also. Now this cell is under turgor. It's under turgor, so it's full of water. And what sort of pressure is being exerted in the actual cell itself? Positive or negative pressure? When we're talking about turgor pressure. Turgor, positive, good, excellent. All right, great. Turgor. Good, moving on now. And I'd like to move on to, I'll just clear that. And I'm going to move on just a couple of slides to this one. All right, so here we revise the concept of water potential, which was introduced last year. And I want you to take a look at this statement here. Okay, but before you do, actually, let's just do a little bit more revision. What's actually meant by the term water potential? What do we mean by water potential? We know the symbol for water potential, something like that, but what does it mean? Water potential. Water pressure. That contributes, pressure contributes to the term water potential. But what is it actually referring to? And like I said, if you're not sure, just say, no, no idea. It has got something to do also with the salt concentration. So the water potential is a function of the solute or osmotic potential, solute concentration or osmotic potential, and it's also a function of pressure. So they both contribute to what we call water potential. Does anyone remember when we talked about free energy of water? Tendency of water to move, it describes that. It describes the tendency or the ability of water to move from one area to another. But let's just focus at the beginning. What do we mean by the free energy of water? Free energy of water. Free energy of water. And if you're not sure, think about it like this. If you had pure water, a sample of pure water, and then another sample of water that had lots of solutes dissolved in it, which one do you think would have more energy in terms of the ability of the water molecules to move? Pure water molecules versus a solution of water that had something dissolved in it. Which one would have more the pure? If you've got pure water molecules, and I'll just draw them as circles, then their properties, their ability, their potential to do work, that is their potential to move, because all molecules have intrinsic motion, thermal motion. The motion, the thermal motion or the ability of these molecules to move around at random is completely unimpeded by any solutes. So we say that pure water has the maximum free energy, and that's given a reference point, a standard value. What is that value? What is the water potential of pure water? Who remembers? What value do we give that? In the units, using the units megapascals, we say that pure water has a value, and it's used as the reference point value of what number? One, you're close, it's 0. This is the reference point, yep, for determining water potential. The reference point is that of pure water, and it's given the number 0 megapascals. So that means any sample of water that has anything dissolved in it will have a negative value, yep, when we talk about water potential, like -1 megapascals, -1.5, and so on, okay, because of the presence of some sort of dissolved solutes, ions, whatever. All right, so free energy of water, and I have lost my, what I was going to say there. All right, so let's now look at this statement. Let's look at the statement. I want you to read that statement. Clear all drawings. If the water potential is more negative within the plant than the surrounding, I think that should be the soil, not cells. The nutrients will move from high solute concentration to low concentration in the plant. What does everyone think about that statement? True or false? This is what I'm asking. If you remember your rules from last year. False? Lachlan saying false? What does everyone else say? Okay, we've got a true, false or true. Okay, so let's do our drawing again. Pure water, no solutes. Okay, and now we've got water, but we've also got solutes. Okay, so we can say here we've got high free energy. Yeah. Here we've got low free energy. In which direction will water move? From high free energy to low free energy, or the other way round? And this is really very much a blanket rule for so many different concepts. The direction of movement from high to low, the water movement. Which way will the water move? From high free energy, high to low. Oh, we've got two different answers there. Okay, water, I think we better go back a little bit. Okay, and just revise a little bit more. Let me just get rid of that. All right, let's go back here and just revise a little bit more. All right, so generally we're looking at the movement. Oh, you've got it. Very good. Yes, high to low. Now we've got 3 correct answers. So we always proceed down a gradient of free energy. Yes, so from high free energy to low free energy. Absolutely correct. So hopefully you're all getting that. So we've got high free energy of water molecules here, lower free energy here. So water will move in this direction. Absolutely correct. All right, so remember, read this slide again just to make sure that you're clear on that. Get rid of that pen. Now let's go back. All right, so now we can look at this statement once again. High solute concentration in the soil. So let's say this was the soil and you've got a high solute concentration here. And you've got a lower concentration here in the actual plant. So do you have, now we're talking about nutrient movement. Nutrients will move from high solute soil concentration to lower concentration in the plant. How does that sound? Regarding what it says about, it's very confusing. It is confusing because this statement is referring to the water potential being more negative in the plant. So what does that mean? If it's more negative, then there's less solutes within the plant. Yeah. There's less solutes in the plant, more solutes outside in the soil. So this is a very confusing, this is a very, very confusing statement, which you're probably best to disregard, and there are clearer statements later on in this lecture that are actually easier to follow. This statement is, I suppose the wording, it contradicts itself in some way. So we can clarify this later on. Yes, I know you're confused. It confused me too. All right, so glad to see that wasn't just me that got confused. Double negative. Yeah, absolutely. Because the first part of this statement is saying one thing, but then the second part of the statement is saying something different. It's contradicting itself. All right, so if everyone would like to just cross this statement off their notes, that would actually be very helpful. Terrific. So I didn't mean to confuse you, but I wanted to know if you were confused. And obviously you are, and understandably so. All right, so let's move on now to slide Okay, slide 38. And I'll just clear this drawing. Clear, get rid of that. All right, so let's look at this diagram here where we're looking at just passive diffusion of molecules through a cell membrane, through the lipid bilayer of the cell membrane. And I think everyone understands this now, that that these molecules, and we'll just consider them to be water molecules, will actually diffuse by the process of osmosis from a region of high concentration to a region of low concentration. And of course, they can diffuse directly through the lipid bilayer itself. What I'd like to clarify is this part of the actual diagram here that describes the concept of equilibrium. So what actually happens at equilibrium? What does that actually mean? Exactly right. Equal movement of these randomly moving molecules of water, equal movement in both directions. Okay, so when equilibrium is achieved, that is, you have the same on both sides of the membrane, same on both sides of the membrane. This doesn't mean that movement stops, because remember, these molecules keep moving around at random, so they'll continue to cross the membrane. These ones will continue to cross the membrane, but at equilibrium there is no overall movement or net movement of the molecules just to one side of the membrane. It's equal movement on both sides, back and forwards. All right, good. Everyone clear on that, on the concept of equilibrium? Equilibrium doesn't mean it stops. Excellent. Good. All right, next. Oh, and before we go on, just thinking about the lipid bilayer of the cell membrane once again. Now, you all know that the interior, so the phospholipid tails that position themselves towards the centre of that membrane, you'll remember that they are highly hydrophobic. Yeah. So would you expect nutrients such as ions to be able to cross, just freely cross, the hydrophobic interior of a cell membrane? What would you expect? No. No. That's right. Water molecules can cross, even though water molecules are polar. They're quite an exception. Hydrophobic substances can cross if they're small enough, but ions, charged species, would be repelled by this hydrophobic interior of the membrane. So ions in the form of nutrients, or rather nutrients in the form of ions, don't just cross membranes. Okay, there's other mechanisms required for nutrients to enter cells, and we'll come to that in just a minute. All right, let's move on now. Slide 47, that's all straightforward. Okay. All right, I like this image. Now let's picture the movement of water, just the overall movement of water from the soil solution surrounding this root into the actual root, without worrying too much about the different transport pathways. I just want you to really focus on a particular concept here. Okay, so firstly, let's look at this structure here. And what do we refer to this structure as, this tissue? And it's shaped like a star, in dicots anyway. Right in the centre, what do we call that? That star-shaped structure in the centre there. What's that referred to as? Everyone knows this. You might not know that it's shaped like a star in dicots. So it's right in the middle there. And this is where the water will end up. The xylem. That's it. That's the xylem, right in the centre of the actual root. All right, so you can see here water movement, okay, indicated by that red arrow there. But what I want you to imagine, because this doesn't really come out clearly, imagine that the water movement is occurring everywhere, right through just about every part of that route. Water is entering by every available transport route. And it's all being focused or concentrated in this central region, into that xylem in the centre. Okay, so I'm not going to keep drawing arrows, but you can imagine. You think about that central point, all the water is gravitating towards that central vascular, that vascular tissue right in the centre of the root. Why is that happening? From what you've seen in your first two lectures, lecture 2 and lecture 3, why is this happening? Why is it all the water in that surrounding soil solution, why is it gravitating towards the centre of the root, towards the
pressure?
xylem?
Pressure.
Pressure? What's setting up that pressure? What's setting up that pressure? Why? It's something simple, so don't overthink it. There's a reason why the water is gravitating. Yeah, that's concentration. Who said concentration? What about
Not me.
concentration? Someone said
Not
it?
me.
Not you? Someone mentioned concentration. Tell me more. Concentration of nutrients. Yes. You can tell me a little bit more. What's happening with concentration of nutrients? That's driving that movement of water right through that root into that central region. There's a concentration gradient, isn't there? Fire in the roots. Something like that. Yeah, a concentration gradient is being set up. Yes, setting up a gradient. Exactly right. So we know that water is moving in passively. There's nothing actively pumping the water into the roots, nothing actively pumping the water up the xylem. But we're not even going to the xylem yet. We're just trying to imagine what's driving water movement into the roots. All right, so someone's mentioned a gradient, a nutrient concentration gradient. Okay, so the concentration gradient is set up so that the concentration of nutrients is the highest here in the xylem, a little bit less here, a little bit less there, and so on as you move out towards the exterior of the root, the outside of the root. So here's that gradient, that concentration gradient, which is established by movement of nutrients into the root. And because it's all being focused here, it all eventually focuses in the xylem, then that is what enables water to follow. Yep, think about it once again in terms of concentration of solutes, or you can think about it in terms of water potential. If you're concentrating the solutes here in the centre, yep, setting up a gradient. If you're concentrating solutes here, so I'll put higher concentration in the centre, lower concentration to the outside. So what we can say? We have a lower water potential in the soil solution that is more negative. Think about it like that, maybe -1 megapascals. Yeah, sorry, maybe -0.5, maybe -0.5 megapascals, whereas here it's actually higher solute concentration. So it would be a more negative number here. Okay, is that making a bit of sense? A little bit of sense to you? Okay, because we can't really imagine what's driving that water, why the water is moving into the root, unless we understand what's setting up that concentration gradient in the first place to enable that diffusion of water, that movement of water by osmosis. Okay, moving on now. All right, pathways into the root. So we're just talking about water movement once again. And once again, let's drive this point home. If we have a high solute concentration here in the center of the root, in that xylem, and then we have a gradient of concentrations through all these cells, and then a lower concentration of solutes in the soil solution, then water will move in that direction. So what does that mean? High free energy in the soil solution, lower free energy of water in the xylem because of the increased concentration of solutes. And that explains why water moves into the root, into the center of the root. All right, so let's look at the different pathways on this slide and on the next slide here. So what do we mean by this term here, the apoplast? What's your understanding of the apoplast, which is shown here in blue, in light blue? Don't know. Anyone? Anyone familiar with the apoplastic pathway? No? Anyone? The apoplastic pathway. So water can come in to the root cells by every possible means of entry, every possible route. One route is the apoplast. Okay, so it's indicated here, the apoplast. So what region or regions are we talking about within the root? No one? Okay. All right, so this region here is referring to that space between the plasma membrane of the cells and the cell wall. It's also referring to the cell wall itself. So water can move via the cell wall, through the cell walls from cell to cell. Within that space between the membrane and the cell wall, and also if there's any spaces between cells, water can also move in that way. So that's basically the apoplast. Yep, all the regions in between the cells are between the plasma membranes, the plasma membrane of one cell and the plasma membrane of the neighbouring cells. All right, water. Does water move directly through a plasma membrane into a cell? Yes or no? Yes, it does. Water can move, can diffuse directly through a plasma membrane. That's all right, no problem. As I said earlier, even though water is a polar molecule, it still does diffuse directly through the lipid bilayer of a plasma membrane. All right, there are also transport proteins within the plasma membrane that are selective for water. They're called aquaporins. And I'll just type that for you. Aquaporins. So these are transport proteins that are much faster at moving water into each cell than simple diffusion alone through the membrane. So that's another way. Okay. I'll just clear that. All right, what's happening here? So let's say water's come in through the plasma membrane or through the aquaporins, and it's coming through this passage here into the next cell. So from the epidermal cell into a cortex cell. What's this passage here called? Who remembers this passage? These very, very fine tubes that connect plant cells. And these are completely absent when you're talking about animal cells. You only find them in plant cells. Who remembers what those fine passages are called? Between the cells, starting with P, letter P. Those fine tubes, they're bounded by plasmodesmata. Thank you. They're plasmodesmata. There are thousands of these in each cell, connecting or joining the neighbouring cells. Thousands of these. Very good. Good work. Plasmodesmata. Remember that? Oh, and here it is here. I didn't even see it. But of course, Laura didn't see that. She just knew that. Very good work. All right, plasmodesmata. So, of course, water can move through these channels as well from cell to cell. Water can move through the apoplast, can move in between cells. So you can see that every possible transport route can move water from cell to cell or in between cells, and until we get to, of course, this Casparian strip found around the endodermal cells of the endodermis. And then, of course, any water that's come in through the apoplast is forced to move in through the cell itself. So this is a concentrating mechanism here. And then from there, it goes through the other cells and enters the xylem. All right, so let's now put names to those various transport routes that we just saw. Let me just clear that. But before we do, just let me check something else that I wanted to talk to you about. Okay. All right. What would happen, do you think, if you over-fertilised your plants? Over-fertilised. So we're in the soil over here. And you got the concentration wrong. Over-fertilised. Yes, that's possible. Yeah. Water the other way. Water the other way. Yeah, absolutely. If you really overdid it, in terms of your inorganic fertiliser, too much organic application of the organic amendments, absolutely you would reverse the concentration gradient, wouldn't you? So think about it. If you suddenly had a very high solute concentration in the soil, very high, higher than you have here at this end, then you're going to reverse the movement, the direction of movement of water. You'll have water actually moving out of the root cells into the soil. What would be the visible effects of this on the plant? What would it look like in terms of physiological signs in the plant? And keep these things in mind for your plant physiological management plan, Because remember, it's hypothetical. So you could say, oh, I accidentally applied fertiliser at this rate instead of this rate. Then you can talk about these processes. Change colour, yes. Dye in extreme circumstances, yep. Or if you didn't do something to manage it, absolutely. Yep. What might you think is happening if you didn't realise? They look crook, yes. What might it look like is happening to the plants? So think about it. If water's actually flowing out rather than flowing into the roots and up, the water's coming back out into the soil. Dehydrated, yes. So what happens to the leaves if the plant's dehydrated? Wilt, exactly right. So if the water's been pulled out into the soil solution, because you've got those high solute concentrations, the plant takes on the appearance of drought stress. The plants will look like they need watering because the plants start to wilt. Yeah, drought stress. Okay, so that results from that outward movement of water. So the wilting obviously is due to the loss of turgor, not enough water actually moving up the plant itself. All right, so when we talk about water potential itself and field capacity of your soil, that obviously depends very much on your soil type. And of course that's a function of the size of the surface area of your soil particles. If you've got large soil particles, which you would expect in sandy soil, then the surface area of soil particles is less than if you had that very fine clay particles. You have increased surface area. So when we talk about field capacity, it varies, but we can think about a water potential of about negative 1.5 megapascals. Would be very rough approximation, as I said, and that depends on your soil type. Field capacity would be about negative 0.01 roughly. Once you get down to negative 1.5, then you actually start approaching that wilting point. Okay, so we have a question. So could be not enough water, but not really. So you could be watering enough, but overfed. Absolutely. So what appears to be not enough water, or water stress or drought stress, however you want to describe it, could actually be a problem with too many nutrients in the soil. Concentration of nutrients is too high. Because think about it, how often would people fertilise just as a routine application without testing first what the concentration of nutrients actually are? How does one deal with that? So how would you manage a soil that you know has been over fertilised? How would you manage that? Any ideas? Water heaps, yep, dilute. You've got to actually try and leach out the nutrients, and to do that you have to keep applying lots of water. What if that water's not available? So what if you didn't have irrigation to be able to leach out those nutrients? How would you know? That's right, how would you know that you've over-fertilised? So you've just applied some fertiliser, and then what happens within a few days, you start to see the wilting effect and you start to see injury to leaves. The roots are becoming injured already. So you would assume that there's a problem with your fertiliser application. But the best way to know is through soil testing. Just basic soil testing before you apply fertiliser. Yeah, soil testing, absolutely. So yeah, trying to leach out those nutrients is really important, but it means that you have to be able to apply a fair amount of water. I don't know what the amount is, but I know that you'd have to have irrigation available to do that. Another way to try and, it would be a lot of water. Another way to try to get rid of nutrients from the soil is to plant some other plant species that would obviously take up those nutrients from the soil. But obviously, if you've already got a crop in place, that's difficult to do. That's probably more practical in the sense of a vineyard to be able to do that, to plant some other sort of crop. Yeah, so now you can understand mid-row crop. Yep, absolutely. Right, let me just clear that. I can never find the clear all drawing and move on. And we'll just move on now to the next slide. This is a great slide because it shows very clearly the different routes that water can actually travel through a plant. So let's just revise these different routes. So transmembrane route is one. Okay, directly through the membrane of one cell, cell walls, and through the membrane, through the lipid bilayer. And what's this called? Transmembrane route. Okay, next one, we have the symplastic route. And the symplast, here we go, also travelling through the cytosol of the cell, but to reach the next cell, water is actually moving through the plasmodesmata. There we go, in blue. And then the final one, in purple, we can see the apoplastic route. So, of course, through the cell walls, so not travelling through the actual cytosol of the cell, through the cell walls, in between the plasma membrane. There's a little space in between the plasma membrane and the cell wall where water can travel, and even in between cells if there's any spaces there. So as I said, any available route, water will travel into the actual, the cells of the root heading towards the xylem, assuming that you haven't over-fertilised, which of course I've done before. Okay. Everyone clear? Any questions so far? Any questions? All happy with that? Terrific. All good. Excellent. Lecture 4. Oh, okay. So this was to remind you, just take you back to living systems where we have... let me clear this, clear all drawing. Here we go. So we're looking at the geometry of a water molecule. And here it shows it, oxygen surrounded by 4 groups. Here we go. So it looks like a tetrahedron, but it's more compressed. So it's actually, the molecular shape is actually that of a bent molecule. And that's when the 4 groups around the atom include 2 lone pairs. Okay, now let's move on to lecture 3 and nutrient uptake. We're all drawing. Right, so I'd like to go to slide 16. Let's see what we've got in slide 16. All right, here we have an illustration of facilitated diffusion, and I assume everyone's up with the 2 major classes of transmembrane proteins that facilitate this type of diffusion, called facilitated diffusion. So what are the 2 major types of proteins, just general classes of proteins that we can see on this slide? So of course this is passive diffusion of molecules, but it's facilitated diffusion because the movement is facilitated by these proteins that are embedded in the plasma membrane, or in fact in other membranes as well, not just the plasma membrane. Enzymes, not quite what I'm after. So two major classes of transport proteins, and the answers are actually on this slide. Okay, two, and you might remember this was actually on the final exam last year. Carrier proteins, yes, carriers. So that's one down here, carriers, and what's the other type? So we've got carrier proteins and one more type, one more class, I should say. There's carriers and something else. And now you're just taking a stab in the dark. Carriers and channels. Who remembers that? Carriers and channels. Okay, what's the difference? Difference between carriers and channels. Channels, so that's a channel. And here's an example of a carrier. Channels open pathways. Yeah, so you can say that channels simply have open and closed states. Carriers are specific. Yeah, channels can be specific as well. What about the rate of movement? Which one's faster? The channels faster or the carriers faster? Channels are faster. Channels simply open. Channels will simply open, and the molecules can simply diffuse through. Yep, along a concentration gradient, down a concentration gradient. Carriers tend to be a bit slower. Because they will bind. They'll bind something on one site that will enable a change in conformation of the carrier protein, and that results in the carrier opening on the other side and releasing that molecule to the other side of the membrane. Yeah, absolutely. Someone's gone or someone's joined us. Yes, that's correct. Channels open and close. You're talking about carriers, that's right. They're the ones that actually have to change conformation. So they tend to be slower, yep. But they're both still examples of facilitated diffusion. All right. Let's move on now to lecture 17. Sorry, slide 17. Lecture 17. All right, so what you can see here in this figure, drawing. So what we can see here is an overview in this figure, an overview of many of the different transmembrane transport proteins that have actually been studied, that have been, studied in some way. There's some experimental evidence to suggest that they exist and to support their function. Now, I'll just enlarge this a little bit. Can I move it? Yes. Just move that out of the way a bit. All right, so here we can get a good look at the different types of transport proteins. And I need to actually move that over, which I can't. That's a shame. No, I can't move it over. That's okay. All right, so let's look at these different classifications of transporters. So when it comes to movement of nutrients, tell me what sort of nutrients we're talking about. Movement of nutrients into the root to set up that concentration gradient which enables the passive movement of water also into the root. Yeah, I'll try, but it seems to then just go to one side. Let me try it again. Just get rid of that. Oh, here we go. It is moving, but I can't move it over any further. Is that better, Amelia? All right. I'll just move this out of the way so you can see. All right, so when it comes to movement of nutrients, what are we talking about? Give me examples. Everyone knows what nutrients plants require. So start typing away into the chat box. Nutrients. Nitrates, yes. More. NPK. Yeah, that was an easy way of doing it. Yep. Yep. Absolutely. What else? Very nice. Laura. Magnesium. Calcium. Yes. Fantastic. Any more? Etc. No, not etc. Boron. Come on, everyone can think of something. And there's plenty of examples even on this slide here in this figure. Etc. Okay. Magnesium. All right, so how do these nutrients get into root cells? Now, as you can imagine, there's lots of different transport proteins that facilitate the movement of nutrients. Let's look at some of them, some of the categories and some of the specific transport proteins that have actually been classified. So let's start first with what we're seeing here. So this is the plasma membrane on the outside. Can I go up a little bit? Yeah. And in the centre here, we have that very large organelle called a vacuole. Does anyone know what the membrane surrounding the vacuole is called? It has a special name. The lipid bilayer membrane that surrounds a vacuole. And this should definitely be in the exam because you can't do plant phys and not know the name of the vacuolar membrane. Mitochondria? No. No. No, you might not have learnt this yet. Actually, I don't think it's mentioned at all. It is a vacuolar membrane, but it has a very special name. Here it is, tonoblast. Tonoblast is the vacuolar membrane. Write that down, commit that one to memory. All right, so you can see the presence of lots of transport proteins in the tonoplast as well as on the plasma membrane itself. Let's just take a look at some of these. And so we'll start down the bottom. Here we go. We've got some channels, which we just referred to on one of the previous slides. So we've got potassium, inward potassium channel. We've got outward potassium channels. Here we've got an example of a channel that doesn't have what we call high affinity for a specific nutrient. They're called low affinity channels, which mean that they can transport 2 or more different types of ions or solutes. So this one can transport both potassium and calcium and sodium. So that's an example of a low affinity channel. Here we've got calcium channel and another one there. So there's some channels, and here's some channels on the tonoplast as well. All right, let's go up the top and look at those green ones. Symporters. Symporters. What do we mean by a symport? I'm testing your memory, your memories. Symport. Who remembers this one? So remember we had symporters and antiporters. Specific ports, they are specific, but what distinguishes a symporter from an antiporter? Sympathetic, no. No, symport. All right, let's look at an example if you're not too sure. Okay, let's look at this one here. So this Symport protein here, you can see it's transporting 2 species. But it's transferring 2 in the same direction. We've got protons coming in, and the movement of those protons, the hydrogen ions, is also accompanied by the movement of nitrate in the same direction. So that's what we mean by Symport. Movement of 2 substances or 2 species in the same direction using the same transport protein. So they're symporters. Here's another example here. So you can see again, we've got a proton, movement of protons into the actual cell, and that movement drives the movement of, what are these ions here? What do we refer to them as? PO4 3-. What are they? Very important nutrient. And someone mentioned it before in an abbreviated form. P. Yes, so these are phosphate. These are phosphates. So this is how P is brought into the cell. Here's another example. Here's a different one. Movement of sucrose is driven by the inward or the influx of protons through another symporter. Here's an amino acid, proton amino acid symporter, a proton peptide symporter, and here we come to an example of an antiporter. So what's the difference here that you can see with an antiporter? What distinguishes an antiport transmembrane protein from the symporters? 1 in, 1 out. Perfect. Okay, so you can see that here, antiporter, protons coming in, and that enables the movement of sodium ions in the opposite direction. So that's what we simply mean by an antiporter. All right, now you can also see lots of examples here on the tonoplast, so the vacuolar membrane. So we can see calcium transport, magnesium transport, sodium, nitrate once again, lots of different examples here. And here I like to test you once again. Hexose, a hexose antiporter. Hexose proton antiporter. What do we mean by hexose? 6-chain sugar, 6-carbon sugar. More specifically, 6-carbon monosaccharide. Yeah, you can say sugar, that's fine. Okay, so that's hexose, and we can also see a sucrose proton antiporter here. Okay, so lots of examples there for you. So this is an overview. This is simply an overview. What I'd like to really focus on is what you would expect to see in root cells, what sort of transport proteins in root cells. Okay, so I think I've finished with that one. Okay, here we go. This is one of my favourite images. So have a little bit of a look at that to get your bearings. So this lower section is the root, and then we've got a little bit of the stem in here, and the top region here is the leaf. So I want you to focus your, isn't that fantastic? Really well done, whoever put this together. Focus your attention on the root, on what's going on in the root. We're going to look at, and this is wheat. This is what's been discovered in wheat roots in terms of transport of nutrients. So have a look at the transport proteins there and what they're transporting. And I want you to tell me what the focus is here. Okay, what are the nutrients that this particular paper was focusing on? Nitrogen, in what form? Yes, absolutely nitrogen. In what form? In what ionic form? Nitrates, correct. and one other. So these are the nitrates, the all-important nitrates, and even more important, what's the other form that plant roots loves to take up? No, not quite, but we'll come to that. Keep that thought. Ammonia, yes, ammonia, but let's say it properly. Ammonia. So you're close. What is this? How do we? Ammonium. Correct. Good. Let's not confuse ammonia, which is NH3, with the ammonium ion. There. If you guys get to third year and do your research projects and say ammonia instead of ammonium, I'll be disappointed. Okay, so that's what this paper is actually focusing on, and the experiments they did on net ammonium and nitrate fluxes in roots of wheat plants. All right, so what do we have here? In yellow, we have a nitrate transporter, which obviously transports a nitrate from the soil solution into the root cell. Here it is. Once it's inside the root cell, what happens to it there? Okay, nitrate can be, one, transported through the root cells, through the stem cells, and into the leaf, into cells of the leaf. What happens to it then? It is changed into nitrite, NO2 minus, and then very rapidly converted from there into ammonium ions. Why? Because ammonium ions are very easily assimilated into amino acids. That is, they're incorporated very easily into amino acids to make proteins. Okay, so that's one fate of the NO3 minus, the nitrites. But of course, root cells also require nitrogen. Root cells require all the nutrients. So some of the nutrients, of course, remain within the cells, the root cells that they're being transported into. All right, so within the root cells themselves, nitrate converted to nitrite, and then immediately, because nitrite is very toxic, into ammonium ion and converted into amino acids to be used in the synthesis of proteins within root cells. All right, so that's that. Let me clear my messy drawings. Oh, and of course, amino acids can also be transported into other parts of the plant. All right, so that's the nitrites. What about the ammonium ions? Transported in to the root cells. Now, ammonium ions can be directly assimilated into amino acids. So you can see why they're so important. The plant finds it very, very easy to incorporate them directly into amino acids. Okay, and each of these changes from one form of nitrogen into another is catalyzed, of course, by an enzyme reaction. And one of the important enzymes that we find in this process is called nitrate reductase, or nitrogen reductase, NR, abbreviated to NR. All right, so ammonium, still on ammonium ions. Are ammonium ions, would they be transported to other parts of the plant in the same way that the nitrates are? Would they be transported? The answer is actually no. They're not transported. They are highly reactive. They are themselves quite toxic if they're not very quickly assimilated. So they don't tend to be transported through the plant. Nitrates are, and of course, amino acids are. All right, now you might see, you might have noticed these reverse arrows, these dotted reverse arrows, which indicates that you can lose valuable nitrates from the root cells back out to the soil. And in a similar fashion, all important ammonium ions can also be lost. So these nitrate and ammonium transporters permit both influx and efflux of important nitrogen ions. All right, so what causes that? What would cause loss of nitrogenous ionic compounds from root cells? And here's the clue for you. Here's one clue: water stress. Absolutely. So when wheat plants are suffering from water stress, yep, gradients, yes, absolute gradients. When there's water stress, then that will actually reverse basically the function of these transporters and causes loss of nitrates and ammonium back into the soil. Absolutely. There's also another negative feedback regulation, which actually comes from here. So when carbohydrates are being synthesized in the leaves, and they're obviously transported around the plant via the phloem. If the carbohydrate concentration builds up in the root cells, then this also will reverse the movement of nitrates. Will also reverse the movement of nitrates so that there is efflux of those ions back into the soil. So there's lots of these different regulatory mechanisms: positive regulation, negative regulation, and water stress would probably most likely be only one environmental stress that causes root cells to lose nutrients rather than move nutrients into the cells. So very, very nice image. I hope you agree with that. So that's one example. Let's look at another root-specific example. And let me just clear that. Yes, so too many carbohydrates. Not so much a problem as such, but it's just a way of making sure that the plant's not expending energy on something that it doesn't need. The plant's got lots of these different regulatory mechanisms. So if the plant's got plenty of can make plenty of carbohydrates, it's got plenty of nutrients, then why would it expend energy on actively moving nutrients into the roots when it's not actually required? Yeah, absolutely. Less energy all the time. Certainly does. That is a law of nature. It's just one of the fundamental laws of nature, to go from high energy to low energy. That theme seems to come up all the time, regardless of what you're discussing. All right, get rid of that. And let's move to the next example that I've got for you. All right, so here we have a schematic of different nitrate transporters that have actually been studied. So second law. Yeah, second law. I don't remember if that's a second law or second law of physics. Thermodynamics. Thank you. Yes, that's what I meant. Absolutely. All right, so let's look at what we're seeing here. So quite simply, we have a root hair, which is right there, this elongated epidermal cell. So obviously the soil solution's around here. Then a cell of the cortex, a cell of the endodermis, and the corresponding Casparian strip. And then we've got some parenchyma, xylem parenchyma cells, and then cells of the xylem towards the left here. All right, so what we can see here are these little boxes, these little circles that we find on the plasma membrane of the root hairs. Plasma membrane, and also we'll see some of these are also found on the vacuolar membrane called the tonoplast. Terrific. So what are those transport proteins? Those transport proteins which are specific for nitrate. Here they are. They're indicated here. One's called NRT12, and then we've got NRT21. Then only on specific cells, in the plasma membrane of specific cells, these black boxes appear, and these are different nitrate transporters, NRT11, NRT18, don't worry about the names. The point of this slide is that there are specific nitrate transporters localized to specific cell types, and they all work together in transporting these particular nutrients from one cell to the next, and then finally in the loading of the nitrates, as you can see here, into the actual xylem itself. There you go. So I'm not sure what plant species was being studied here. That doesn't matter. All right, another example. A little bit blurry, this one. And this is a schematic of some nutrient transporters that were studied in rice, in roots of rice. So you can see here they're concentrating on manganese, yep, and silicon. And they isolated and they categorized these transporters here that were specific for these particular nutrients. Okay, there's something else that I wanted to say here. Oh, yes. All right, so the endodermis you're familiar with. Here's the endodermal cells, and this is supposed to represent the Casparian strip. But in rice, there's another Casparian strip, and it's found in this cell layer here. So this is called the exodermis. This is the layer of cells immediately beneath the epidermal cells. And they also have a Casparian strip, which is quite interesting. So that basically puts a stop to any nutrients, unwanted nutrients that are actually coming in through the apoplastic route, and it forces them through the symplast before they even get to the endodermis. But remember, any nutrients that are coming in with water in the apoplast, they can only actually enter the cells, or mostly enter the cells themselves into the cytoplasm through nutrient transport proteins. And you can see that here. So here you've got manganese, for example, that's coming in through the apoplast here, in between cells or through cell walls, for example, but it needs to actually come in through a transport protein to get into the actual cell itself, into the cytoplasm of the cell. All right, I think I've got a couple of examples. Now, this is also very interesting. The influx of nitrate and quite likely the influx of any nutrient into root cells depends on various environmental factors. What do you think those environmental factors would be that would determine whether nitrate, for example, is moved into the root cells, or whether it will be moved out, and at what levels? Environmental factors. Moisture, yes. Temperature, yes. And what's being shown here? The time of the day. Yeah, absolutely. Time of day even has an effect on how much nitrate is being imported, actively imported into the root cells. And you can find lots of examples like this if you do a bit of a literature search. So this is interesting. If we just look at this axis down here, time, where time 0 would probably represent dawn. And then here, so where we've got this dark area here, this would be night, so day and night. And you can see clearly, and this depends on the concentration of nitrate that's available. So this is quite a high concentration, 5 millimolar versus 100 micromolar. So when there's a high concentration of available nitrate, let's look at what happens. So at dawn, daybreak, very high influx of nitrate into root cells. Then the amount of influx reduces. Then for some reason, there's another peak here towards the end of the day. And then during the night, the level is probably at its lowest. But it's still, the root cells are still moving nitrates into the cells, but at lower levels. But the movement is still occurring. So that's when there's lots of nitrate available. Look at what happens when there's much lower levels of nitrate available. You don't get quite the same peaks occurring during the night and day cycle. So I find that interesting. I'm not sure how to explain that, but I'm sure if you were to refer to this paper, you would probably get a good explanation, or at least some theories as to why that's occurring, why you get this sort of these different peaks during the day and night. Okay. Solar energy. There's one possibility. All right, and finally, this is our final slide for tonight. And I thought it very useful to consider how a plant tolerates toxic elements in the soil. Toxic elements in the soil. What does it do to tolerate this? And obviously, if this is a sign of tolerance, if these are the mechanisms that are involved of tolerance, then obviously plant species that are sensitive to particular toxins in the soil must lack these particular mechanisms. So this is a study that was done in Arabidopsis, and it's looking at aluminium tolerance. So you know that aluminium ions, pictured here, are toxic, and they are a problem in certain parts of Australia. So plants don't seem to have any need for aluminium, but they need to be able to manage those ions in some way. And actually, I was pretty happy when I found this particular example, because these transport proteins that are involved in aluminium tolerance, ALMT1 in particular, I actually studied those for just a few weeks, but many, many years ago. I studied the ALMT transport proteins, but in white clover. The study was being done in white clover, not Arabidopsis. But I wasn't actually studying the transport proteins themselves, but rather the genes that encode these transport proteins. So let's just simply look at the mechanism that's being proposed to enable tolerance to these toxic species in the soil. All right, so here's aluminium in the actual soil itself. And here we have an epidermal cell, and of course an example of a cortex cell, endodermis, once again with its Casparian strip, pericycle cell, and the xylem here to the right. All right, so aluminium ions can move via the apoplastic flow in water, in between cells or through cell walls, but of course not enter directly into the cytoplasm of these cells. Now, that movement into the apoplast actually triggers this transmembrane protein called ALMT1. It triggers it to actually secrete or transport malate from inside the cell to the outside of the cell. And what malate does is that it binds up aluminium ions. It binds them up, and in that form, they can no longer enter. They can no longer enter the cells. They're not toxic to the cells. And there's the mechanism for tolerance through the action of ALMT1. Here's another theory that there's that ALMT1 will transport malate out even into the soil solution, not just into the apoplastic region. And there the malate can bind up these toxic aluminium ions. All right, so that's one mechanism. Let's look at another one. Here we have aluminium ions that are actually being transported by this transmembrane protein called NIP12. Now, what happens when aluminium actually is transported into the cells? Then it will bind, and now we look down here, then it will be bound up by malate to form an aluminium malate complex. In that way, it's no longer toxic to the cell. How does the cell deal with it? It transports it through the plasmodesmata from cell to cell. Here we go, aluminium malate complex, transported through the NIP12 transmembrane protein, loaded into the xylem, and then it's transported up to the leaves, and there it's actually stored in the vacuole in the leaf cells. And of course, the aluminium malate complex can even be stored in the vacuoles in the root cells as well. So that's basically one way. Malate is it's an ester, if you like, an ester of malic acid, a substance called malic acid. Yeah, it probably relates to what viticulture and winemaking students know as malolactic fermentation. But of course, I have no experience in that area whatsoever. Okay, so I think just a nice illustration of why some plants exhibit tolerance to these toxic species in the soil, and obviously other plants must lack these mechanisms. And it was even interesting when I was doing a little bit of work on this many years ago to see that different white clover plants, even though they're exactly the same species, but different individual plants had varying levels of tolerance to aluminium. So obviously that intrinsic basal tolerance, which depends on the genetic background of each plant, is different from plant to plant. You have that variation. Okay, so that concludes tonight. Is everyone still there? Are you all still with me? Yes, terrific. All right, any questions? Now, yeah, I know. And look, I'm sorry there wasn't time really to go through your first tutorial, which is the review, the journal review. Has anyone managed to do the journal review yet? Shoot 1, review of 3 journals, not yet. Okay, well that's good that we haven't had time to go through it. We will come to that and any other tutorials in 2 weeks' time, which will be our next class. So everyone remember there's no online class next week, but there will be the week after. So from now on, every second week we'll have a class. Your job's not that stimulating, Lachlan. Does that mean you need a new job? So any questions from anyone? All right, thank you everyone. Yep, thanks everyone for all your participation, and I'll see you in a couple of weeks. Okay, good night.